Okay, hello everyone. Welcome everyone again for being here today and join us in our brain-body interaction series. I'm very happy to be here today hosting Dr. Pierre Leopold. Before introducing our speaker of today, I just wanted to give you some notes. As for the other talks, you are free to ask whatever questions you want during the talk. You can use the button "Ask a Question" that we have here below, and you can leave your comments as well in the chat. And then people can also vote for your questions. So feel free to ask all the questions that you have. And also after the presentation, after the seminar, we also have meet the speaker time. So we have several seats which have been taken already, but we can also always squeeze in someone who wants to really join us. And so if you do, just drop me a message and I will send you to the Zoom link. So we also have, as usual, a poll, and we would really like you to go ahead and vote for a question, for this question, or for the answer to this question. So today we are interested in knowing more about the technologies that you use in your projects, right? So we have several options regarding what could be interesting technologies to use in this team of brain-body interaction. So feel free to vote for your favorite answers. And I think it's basically this. And then, so I will move forward to introduce you to our speaker of today. So Pierre, as I said, I'm very excited to have Dr. Pierre Leopold today with us. He's been very influential to me, to my science personally. So he has graduated from the University of Nice at a time where a lot of discoveries were being made in the embryogenesis field and also related to the development of the body. And so during this time, he started being very interested in this type of thematics, and he started working on Drosophila, which is kind of the prime, let's say, model organism to study these questions, developmental questions. And he did his postdoc. He was a postdoctoral fellow at the University of California, San Francisco, and he was trained as Drosophila developmental genetics. And so during this time as a postdoc, he tried to find out how cell proliferation was controlled by developmental signals. Okay? And so when he finished, he managed to obtain a starting grant, a CNRS starting grant to return to France. And that's when he started his own lab at the University of Nice. He's currently the director of the Genetics and Developmental Biology Department at the Institut Curie in Paris, and the research director of the National Institute of Health and Medical Research. So when he started his lab, by his own words, he says that logically he went from the question of how cells are organized within organs to the quantitative problem of growth and the production of tissue mass. So during the past years, he has made tremendous contributions to our understanding on how organism growth is coordinated. So he has been using Drosophila as a model system to dissect the mechanisms which underlie the control of tissue growth, mainly the systemic mechanisms. And he has so far been able to dissect specific organ crosstalks and molecular pathways controlling organism growth according to nutritional availability. And more recently, his lab has been focused on the importance of inter-organ communication in the mechanisms of organ growth, coordination, and size determination, which I think is going to be the main focus of his talk today. I'm very, very honored to be hosting Dr. Pierre Leopold today. I'm looking forward for your talk and for further discussion. So the stage is yours.
Thank you very much, Zita. So I'll try to share my screen now.
Exactly.
Okay, so this. Can you see my screen?
Yes.
Okay, and pointer option. And can you see my pointer moving, this red dot moving?
Yes, everything looks fine.
Okay, very good.
Thank you.
Okay, well, thank you very much, Zita, for the introduction. The impressive introduction. It looks like you really went through the work of the lab, and I'm very impressed. Makes me feel more about what we've been doing than myself. And also a big thank you for the invitation and for the committee to invite me to talk. It's a pleasure to be connected again to the Champalimaud, even if it's a virtual connection. It's an amazing place. I have very good friends and colleagues there, so I haven't been traveling for a while, and I'm missing meeting you guys, as many people do. So as Zita said, I will try to talk about what we've done recently on the control of growth and using Drosophila as a model, which we have been doing for the last maybe 20 and something years. And I will focus on research that we have done recently on mechanisms allowing coordination between the growth program of the different part of the body. Let's start with a very, very broad introduction. Growth is controlled mainly by two different ways, what we call intrinsic parameters or intrinsic cues, which are derived from genetic, exemplified in this picture of these two nice twins here. They share the way they love. They share many things, including size and shapes. And so this is a good illustration of how genetics can actually control the growth program. And on the other side here, we have a picture of two flies, two Drosophila adults, which are both males, and they also share the same genome. But in this case, they reach very different sizes at the adult stage. And this is only due to the fact that they have been growing as larvae, as juvenile, on very different food media. So this illustrates how environmental conditions can actually influence and modify the genetic program of growth, and trying to understand the intricate relationship between these two types of influences controlling growth is really a major task in the field. So we've been working on these aspects, the intrinsic and specifically the nutritional control of growth, for a number of years. But I will talk today about something which is slightly different, which has been started a few years ago in the lab, which is actually related to the intrinsic control of organ growth, and it's very nicely depicted here in this early experiment, reciprocal grafting experiment, using two salamander species, a small species, Punctuatum, and a large species, Tigrinum. And so if you take the limb bud of the large species and you graft it on the small species and you do the reciprocal grafting, what you end up seeing after the growth is completed is a limb which has the size of its species of origin. So very early on, that was a very inspiring and crucial experiment in the field of growth, suggesting that individual organs have a way of controlling their size, their final size, which is independent of their environment. At least independent of the genetics of their environment, which is the case here. And I will come back to this distinction later in my talk. So when are we growing, and when do organs grow? Well, in fact, in many species, including us, humans, and also many terrestrial species like insects, terrestrial insects, we have a a determinate growth program, which means that we grow during the juvenile period of our development. And we stop growing at the end of the juvenile period when we transit from juvenile to adult. So basically when we mature, when we acquire sexual maturation and the capacity for reproduction. This is true for human, and we all know that we stop growing at puberty after some kind of a spur of growth spur. And it's the same in insects. Insects grow mainly during what we call the larval period, which is the juvenile period, and then they kind of stop growing. At least the system, has acquired his definitive mass during a stage which is called pupa, pupal development, during which metamorphosis and transformation into an adult is occurring. This is also the stage during which sexual maturation is taking place. So this is actually quite comparable in some ways, and what is even more comparable is that this transition from the juvenile to the adult stages is actually marked by a very strong induction of steroid hormone production. So these steroids in vertebrates are called the sexual steroids, and in invertebrates, in insects in particular, they're called ecdysone. I mean, this is the main steroid is ecdysone, it's called ecdysone. So what becomes visible here is that if organs grow in an autonomous manner during this juvenile period here, there must be a way of controlling that everything's going okay before the system is actually stopping growth and is turning into the maturation stage, right? And this has been so to specify what I'm talking about. This is here the life history of Drosophila, which starts with an embryo and then develops as a larva and transforms into this prepupa and then pupa, during which metamorphosis occurs, and then there is emergence of an adult here. What I'm talking when I'm talking about this ecdysone peak is this peak here in blue, which corresponds to the transition between the end of larval growth period and the start of the maturation period. So basically prepupa and pupal development leading to adult. This is just something that I will come back to during my talks. It is the place here, the ring gland is the place where ecdysone, where the steroid hormone is produced. This is an endocrine organ, very, very closely attached to the brain through axonal projections from neurons coming from the brain. And I depicted here two neurons, which are called PTTH neurons. They produce a hormone which is needed to activate the production of ecdysone by the ring gland. And I will, again, describe this later during my talk. So this is how this transition is marked, with this peak of steroid production, which is taking place in this specific organ here, the ring gland. So for quite long, we knew that there was a connection or a coordination between the organ growth program and the timing of maturation, and that was coming from experiments made in the late 70s, early 80s. Mainly by Alan Chern, Pat Simpson, and Peter Bryan, where they could actually modify the growth program of these little structures here, which are called the imaginary discs, which are the precursors of the adult structure. So these structures are actually developing, growing during the larval stage, when the tissues are expanding, growing under the frantic feeding behavior of the larva. And so these discs grow, and then they will transform into adult structure during pupation, during metamorphosis. And so if you perturb growth of these structures during the juvenile period, the normal transition that takes place at the end of juvenile and that pushes animals into pupal development does not occur at the right time. For instance, you can either make an injury to these discs and they will be able to repair, but the time for repair is actually prolonging the juvenile period and extending the juvenile period until the repair is finished. And only at that period, the transition from juvenile to pupa to pupal stage can occur. The same happens if you create a disc which cannot stop growing, and you can do that by, for instance, perturbing apico-basal polarity. This will lead to a structure which is close to neoplasm. So it's what we call neoplastic discs, and these discs will continue growing, and they will not stop growing, and therefore this will inhibit the normal transition from juvenile to pupal period that occurs at the end of 4 days of larval development. Okay, so in both cases, what was evident here is that there was some kind of a feedback mechanism which was able to delay the larva pupa transition according to the growing state of the imaginary discs. And this led, in particular, Pat Simpson to have this very visionary sentence written in one of her papers in 1980, which is that we would like to propose that there is a direct relationship between growth of the imaginary discs and the timing of preparation, and that discs that have not completed a certain amount of growth are able to inhibit preparation, possibly by, in some way, preventing the release of a small peak of ecdysone at the beginning of the third larval instar. The nature of this feedback is unknown. So that was really, really inspiring, and it led us to, and so that was the mechanism. In fact, a factor was produced by the ill-growing tissues that would prevent the normal transition from juvenile to pupa, to maturation. Okay. So we decided to go and screen for this factor, and we use Drosophila genetics for that. And we found one factor that was named DILP8 by our colleagues in Alicante at that time. DILP8, I will come back to that. And so indeed, when removing DILP8 production from the ill-growing tissues, we could actually restore the normal timing of the juvenile to pupa transition. So we could actually rescue the delay normally induced by the repairing or by the neoplastic growth, right, in both cases. So DILP8 stands for Drosophila insulin-like peptide 8, and this this name was given by our colleague Alison Gontiro and Maria Dominguez, and that was due to the fact that they could find a very nice series of conserved cysteine in the sequence of this new protein, which is typical of the superfamily of insulin, IGF, and relaxin hormone. Okay, and therefore, because already 7 DILPs were known, had been identified in the fly genome, this new one was named number 8. Okay, nice paper in Science. We could find that actually it was sufficient to induce DILP8 in the discs, to ectopically express DILP8 in the disc in normally growing animals to delay the larva to pupa transition, right? So in accordance with the role of DILP8 as a major factor controlling the timing of the transition, and this is shown here, where what we depict here is the 50% time of preparation in a population of larvae. And what we see here is that if this is a control timing, and if we overexpress DILP8 in the imaginary discs, what we see is a delay by 2 days, actually, in the time where 50% of the animals have preparated. The reverse is not really true. So basically, if we remove DILP8 from the genome by doing a KO over deficiency in this case, we don't see much of an effect on the timing of preparation of these animals, indicating that DILP8 is not limiting for fixing the time of preparation, but if it's overexpressed, then it becomes it creates a delay. Okay, so then we went and looked for the receptor. Although it was identified as a possible insulin-like peptide, it didn't look like it was close enough from the other insulins that it could work through the insulin receptor. So it was possible that it would work through another receptor, and we looked for it using genetic ways. I will not go into the detail, but basically we found that we and others actually, and again the lab of Maria Dominguez and the newly emerging lab of Alison Gontiro at that time, our three labs came up with the same finding, which is that dilp seems to be working through a specific membrane-associated protein called LGR3 for leucin-rich repeat, containing GPCR3. So one member of a superfamily of receptors, among which we find in this group C, the relaxin-like receptors. So it was kind of comforting to find that dilp was working through the function of this molecule here, this receptor, which was described as a relaxin-like receptor. So this was shown by the lab of Maria Dominguez to be coupled to trimeric Gs and to activate pKa in order to promote cellular response. Very interestingly, then we looked where this receptor is expressed, and we knew from genetic data that dilp was probably acting on a central relay in the brain, not directly in the ring gland that produces ecdysone, but maybe using an intermediate relay in brain neurons that could connect to the ring gland. And this is exactly what I'm showing here. This is a ring gland, so this is the place where ecdysone is produced. What we know is that if we overexpress dilp, we delay preparation, and this is due to the fact that the peak of ecdysone, which normally comes after 4 days of larval development, is actually not taking place. So it's inhibited and it's delayed. So therefore, we knew that dilp was working through the control of ecdysone production, which is taking place here in this tissue, in the ring gland. But when we looked where the receptor for dilp, LGR3, is expressed, we found a large number of neurons, but we focused on this tiny number of neurons, this small number of neurons here, because of genetic interactions, which are located in this specific region of the central brain, and which project their axons in this direction here. And very interestingly, what we find in this region here, where the LGR3 positive neurons project their axon, is an axonal path or neural path for these PTTH neurons, which I already described to you, which have a very specific path. They go like this, they do some kind of a tour in the contralateral brain, and then they exit the brain and they go and project onto the prothoracic gland. So these neurons produce PTTH, which is a hormone. And by producing and inducing, targeting PTTH onto the ring gland, they induce the production of ecdysone, right? And so what we would see here is that these neurons, the LGR3, the dilp responding neurons, LGR3 positive, could possibly connect with these PTTH neurons. And in order to find, to further suggest that we did this experiment, which is called a GRASP experiment, so a GFP reconstitution across synaptic partner, whereby we express half of the GFP in the PTTH neurons and the other half in the LGR3 positive neurons, so in the blue and the red neurons. And if they are close enough, they reconstitute the GFP, and the GFP reconstitution sites are labeled with green. And so this is not a full demonstration of functional interaction, but at least it shows, and this is what we find here with the GFP reconstitution, a channel. We find many places where these two populations of neurons are close enough that they can reactivate the production of an active GFP. Okay, so the model was the following: upon growth perturbation in ill-growing tissues, dilp was produced at very high rate, would travel through the hemolymph, would reach the LGR3 positive neurons in the brain, which would, in some ways, and I think we still don't know exactly what happens here, interact negatively with the function of the PTTH neurons, therefore preventing the production of ecdysone, and leading to a delay in the transition from larva to pupa. Okay. So this is actually a summary of a whole number of papers that were published quite coordinately in the field. Okay, so we went a bit further and we analyzed one aspect of growth coordination, which became clear after a few experiments that we had done, which is the following. So we can specifically affect, which was the way we actually set up our initial genetic screen to find DILP8. We were inhibiting growth, perturbing growth in the specific region of the wing imaginal disc, which is shown here, which corresponds to the future pouch. The future wing blade. Sorry. So this region here in the wing disc is called the pouch, and it will form the wing blade. Okay. And so we could target growth inhibition specifically in this region. And when we do this, we have a strong inhibition, which is shown here if we look at tissue area. Okay. But very interestingly, if we looking at other discs or other parts of these discs where the growth inhibition is not targeted, like for instance this region here called the notum, or these regions surrounding the pouch, which is called the hinge, or another disc like the eye disc. So these are not actually targeted by the GAL4 system, which we use to drive growth inhibition, right? But if we look at these regions, they are also reduced in size. So this is the total of hinge plus notum surface. We see that it's also reduced, and the eye disc was also reduced. Okay, suggesting that. Okay, I think it's shown here. Yeah, suggesting that by reducing growth in the pouch here, we were generating non-autonomous effects in the hinge and notum region, very nicely shown on this picture here. So you see reduction in the surface of the pouch, but also reduction in the notum and the hinge, but also in the eye disc, right, compared to here. So suggesting non-autonomous growth inhibition. How is this non-autonomous growth inhibition carried? Well, we could show that, in fact, I think I need that. Yeah, growth inhibition was performed by turning down, by silencing a gene and encoding a ribosomal protein called RPL7. We know that this is so. We mimic a minute-like mutation, minute-like effect, but specifically targeted in the wing pouch. And if we do that, and at the same time we remove DILP8, which we know is normally produced in these discs, right? So if we do that, so this is the normal situation where we only remove RPL7 in the pouch. We see coordinated reduction in size of the pouch and the notum and the hinge. But now if we remove DILP8, which is normally produced in this central region of the wing disc here, the pouch, at the same time we do the RPL7 RNAi, the growth inhibition, we rescue the growth inhibition in the rest of the disc, right? So we still keep the growth inhibition in the pouch, but we rescue inhibition in the non-autonomous aspects. So this is true in the hinge plus notum, and I don't show it in the eye disc, but it's also true in non-autonomously in the eye. Okay, so suggesting that DILP8 is required to coordinate organ growth inhibition. Okay. So what is leading to this DILP8 induction? What is upstream of DILP8? We actually found that none of the previous pathways that had been shown to induce DILP8, like the GNK pathway or the Hippo pathway, are involved in this induction in case of growth perturbation. But the new pathway, starting with this transcription factor called XRP1, was involved. And indeed, if at the same time we remove, so this is the non-autonomous, right, so this is the DILP8 mRNA levels, so they are very strongly induced when we do RPS3 RNA here in red. They are not really modified if we turn down JNK, so TAC1 RNAi, nor Yorkie RNAi. But now if you remove XRP1 at the same time, we completely abolish the upregulation of DILP8 in this growth alteration paradigm. So now what happens to the non-autonomous growth inhibition? So it's also rescued when we remove XRP1, as shown here. So suggesting that indeed XRP1 is needed for one, DILP8 induction, and second, non-autonomous growth inhibition in response to RPS3 RNAi. Okay. Interestingly, upstream of XRP1, we found a very interesting gene which is called RPS12. So how did we came across XRP1? XRP1 was part of the different hits we got from our primary screen where we found dilp. And so we didn't know how XRP1 was working, and then we found that it was actually upstream of dilp, which was making a lot of sense. So what's upstream of XRP1? We found through a very nice paper that was published by Nick Baker, a specific protein, ribosomal protein called RPS12, which is a very small, tiny ribosomal protein being part of the small subunit, and the last actually subunit to be assembled to form the small subunit of the ribosome. And so the work of Nick Baker described it as a possible sensor for ribosomal function that would be implicated in minute-induced cell competition. So we tested whether RPS12 could have a role in this interorgan coordination, growth inhibition, non-autonomous growth inhibition, and indeed it does. So look at this. This is the growth inhibition, autonomous growth inhibition in the wing pouch. If we knock down RPS12, we get growth inhibition. If we knock down RPL7, we get growth inhibition. If we knock down both, we get growth inhibition. So this is autonomous growth inhibition, okay? What happens in non-autonomous growth inhibition? So if we knock down RPS12, we don't see this non-autonomous growth inhibition in the hinge notum and eye, okay? We see this non-autonomous growth effect as described, I just described, if we remove RPL7 function. So in the pouch, we see a reduction in hinge and notum size, as well as eye size. But now if at the same time we remove RPL7, we remove RPS12, so this sensor one, we rescue the non-autonomous effect. So suggesting that if the disc doesn't have the RPS12 function, it cannot trigger the non-autonomous growth inhibition, which is normally triggered in response to the minute effect. Okay, placing indeed RPS12 very upstream of the cascade of events leading to a dilp upregulation. And this is what happens actually. If we remove RPS12, we don't see an upregulation of dilp, although we have strong inhibition of disc growth. If we remove, if we silence RPL7, we know, as described before, that we have this upregulation of dilp protein accumulation, as we see here. If we do the double silencing, so RPL7 RNAi, which is this, right, plus RPS12 RNAi, we suppress the upregulation of dilp. So this is what we think is happening in response to ribosomal dysfunction or growth perturbation. We don't know how many of these different growth perturbations lead to the same phenotype. This is something that we have started looking into. But it looks like RPS12, so this very specific ribosomal protein could serve the role as a sensor for ribosomal function. And if this sensor is activated, it leads to the upregulation of XRP1 function, dilp induction, and non-autonomous growth inhibition. Okay, so just to sum up, ill-growing tissue pathway, we have RPS12 probably there, we have XRP1, dilp is produced by the tissue, by the ill-growing tissue, activates LGR3, interacts with a PTTH function, and leads to a delay in maturation, as well as an inhibition of growth. And we know, in fact, that ecdysone is actually carrying both the function of the timer of the transition from larva to pupa, as well as a growth factor, which is needed for the discs to grow. So if we inhibit the production of ecdysone, we have a non-autonomous growth inhibition that is observed in other parts of the discs or other discs. Okay, so that was where we were, but except that very nicely our colleagues Alison Gontiro, Maria Dominguez, and Andres Garelli had shown previously that in a dilp mutant animal, although the animals are viable, they accumulate all sorts of defects, which are the sign of what we call developmental instability. Okay, so I will talk more in detail about this because this is what we have been focusing on recently. What is the role of DLP in controlling developmental stability? So how do we control for developmental stability? One classical way of controlling for developmental stability is to look at bilateral organs. Bilateral organs are embedded in the same environment, developmental environment. They are subjected to the same developmental cues because they are in the same animal, and they are also subjected to the same environmental cues because they are also developing within the same individual. So they can be used as a good readout to look for developmental, let's say, stochastic variation in the developmental program. And in a normally developing individual, we don't observe much difference between, for instance, the left wing surface and the right wing surface. These two wings are quite comparable, and they are actually comparable up to within 1%, less than 1%, which is comparable to the size of our two arms, you know, in us humans. So they differ by less than 1%. So this is what happens normally. So normally these developmental errors or developmental, let's say, stochastic variability is actually corrected for by what we call buffering mechanisms, which allow developmental stability or canalization, depending on whether we talk about developmental variability or environmentally induced variability. Anyway, there's a very simple way of looking at that in a fly, which is to measure the surface of the two adult wings within a given individual, and to do that and to reproduce that in a population. And then we measure what we call the fluctuating asymmetry index. So these asymmetries are called fluctuating asymmetry because sometimes it's bigger on the left side, sometimes it's bigger on the right side. And we can calculate an index for that, which is here, which is calculated here as the variance of the difference between left and right, the normalized difference between left and right. Okay. What initially Maria and Andres and Alison observed is that in a DLP mutant, we observe this variability. We confirm this in the case of the receptor for DLP, so LGR3. And therefore, it looks like the DLP LGR3 pathway could be involved in controlling developmental stability, right? How is this working? So this is the data that we actually published. So if this is a DLP control, so this is DLP heterozygous, DLP homozygous, we see a big increase in fluctuating asymmetry index, so the variance of the differences between left and right wing surface. So it doesn't mean that all animals are asymmetric. It means that in a population, there's a small fraction of animals which show variability. But many animals actually are perfectly symmetrical, right? But the variance of the difference between left and right is actually much increased in the DLP mutants. Same thing for LGR3 mutants, where we see an increase in the variance, right? And now we went further, and we could actually suppress the function of LGR3 in this population of neurons that we had defined that correspond to this subgroup of neurons, and indeed this is sufficient to increase AFA index, suggesting very strongly that this subpopulation of neurons, which are the same subpopulation that are involved in the delay, right, in creating the delay in response to a growth damage or this damage, are involved also in controlling AFA. Okay. So how is it working? What we knew from the damage regulation is that DLP is normally produced in this case by the damaged tissue, injured tissue, right? And it acts on LGR3 and relays to the PTTH neuron, the ring gland, and ecdysone production. What happens in the normal during normal development when DLP function is not there? Why do we get FA, and where is DLP normally produced? Well, two possibilities. In fact, for that we started from the beginning, which is one: do we see the correction for fluctuating asymmetry taking place? Is there a time point during development when it's corrected, or is it corrected during development? So the two possibilities are depicted here. It could be that there are continuous feedback mechanisms that allow the discs to correct for their size while they grow. And so there is no need for a specific time for correction because they correct all the time during their growth, right? They could correct by comparing, or they could correct by using feedback mechanisms within a growing individually growing disc, right? I mean, the result is the same in this case. There's no need to stop and correct. The discs are always on their growth trajectory. Alternatively, one could think of a different mechanism whereby there's a developmental time window during which the correction is taking place. So there is no correction during, let's say, larval growth, and then at a certain time during development, let's say larva to pupa transition, there is a measure and a correction that's taking place, leading to adults of with symmetrical wings. So for this, we looked more closely at this pouch that will give rise to the adult plate, and we decided to measure the volume of these pouches while they develop, okay, to see and to measure on both sides and to compare the left and the right side of the two pouches of the two discs that while they develop, and to see when differences are accumulating, whether they accumulate or whether they never accumulate, in order to first discriminate between these two possible hypotheses. So we did this, and in order to do this, we had to segment the discs at different time during development. And what we observed is that if we take this disc, so for that we were expressing a Jeffrey marker with a GAL4 line, which is driven by the nubbin promoter. So nubbin is a very important gene for the identity of the pouch. So as soon as the pouch identity is actually determined, nubbin is expressed. So we found that if we take the discs too early when nubbin is starting being expressed, the expression is not completely established yet, and then we cannot use this very early time point. So we had to focus actually on later time points, 96 hour, which means mid larval 3 stage, and early pupal stage, as you can see here, when these discs have everted and they start forming the blade, right? And so we did some measurements, and what you can see here is that the error, the measurement error is actually lower than the difference between one left and one right disc, and this is taken early at this time point. So the whole system was actually set up, this whole protocol was set up by a fantastic master student, Fabienne Brucher, in the lab. And thanks to this, we could do the measurement. So if we do the measurement, this is what we observe. So we have basically 3 time points. They do not correspond, sorry, to what I'm showing you. I'm sorry about that. There's a mistake in the representation here. So one is 96 hours, so this time point. One is later, just before the end of larval development, 114 hour, and one is just after the transition into pupal development. And what you can see here is the variability is actually reduced very much during pupa. If you represent this in terms of fluctuating index, what you see is that there's a first drop. It's a minor drop, actually. It's less than a 2-fold drop that happens very late during larval development, and then there's a major drop that takes place very early during pupa, right? And then we did the same measurements, but this time in a Dilp mutant background. And very interestingly, what we found in the Dilp mutant is the following. I'm focusing on the index, FA index here, is that the variability is the same in a Dilp mutant as compared to a wild type at 96 hours. It's the same at 114 hours, but then it's completely different after preparation occurs. So it looks like there's this major adjustment of the left-right size difference between the two discs is mainly operated through the function of Dilp, but not this one here, this minor adjustment. Okay. So that was interesting because in some ways it was for us suggesting very strongly that we were in a mode that was closer to this mode, developmental time window, than the continuous feedback mechanism. Okay, and that dilp was playing a role specifically at the end, after the larval development is completed. That was a big surprise for us, I must say. And then we looked at when dilp was expressed, and very nicely, and this is now known, and Alison has produced a nice bioarchive manuscript where he shows the same, and it's also something that was present in some databases. There is a huge induction of dilp expression very precisely at the larva-pupa transition. It's a stage, very precise stage, which is called the white pre-pupae. And this stage is restricted to a 2-hour time period. And during this 2-hour time period, what we see is that there is a dramatic induction of dilp mRNA levels, more than 1,500-fold. And 2 hours later, it's completely down. Okay, so it's a massive burst of dilp and very very, very limited in time induction of dilp expression. And then we used some kind of a TS dilp allele. So we used a tubulin TS, what we call a tubulin TS genetic system, which is a tubulin GAL4 with a tubulin GAL80 TS system. So basically, it's just like we are inhibiting dilp during a defined period using a temperature shift. So when we're shifting animals at 29, the dilp RNA is induced, and therefore the function for dilp is removed. So if we remove dilp function all during larval L3 and pupa, so constantly, animals are constantly at 29, we see the FA in the adults. This is the control here at 18 degrees, so no FA, this is not significant. If we do a 24-hour pulse of dilp inhibition during larva, we don't see any effect. If we do the pulse of dilp inhibition right at the transition between larva and pupa, this is sufficient to recapitulate as much FA difference between the control and the TS as we see if we do a constitutive inactivation. So from this, everything was fitting with the idea that dilp was expressed at the white pre-pupa, at the YPP stage, and needed at the YPP stage to control for FA. The next surprise was when we started looking at where is dilp needed. So what we did first was to knock down dilp in the wing discs. And by knocking dilp in the wing disc, we couldn't see any effect on the. Sorry. This is not RNA. This is just looking at expression in different tissues at the white prepupa. So if we look at the different tissues, dissected tissue from the larva from the white prepupa at this precise stage, so we have to be very precise. If we miss it by plus minus 1 hour, we don't see dilp expression. So if we do this, we only see expression in the carcass, which is for people who have this dissected larvae, it's the association of the epidermis and the muscles, which are actually opposed to the epidermis and sticking to the epidermis. So we cannot really dissect the muscles from the epidermis, and we can only have both at the same time. But now, if we use a specific GFP marker for dilp expression, which is shown here, we can very clearly see that it's localized in the epidermal layer here, but not in the muscle layer. Okay, and this is shown here. These are the muscles in red, labeled with phalloidin, and this is a dilp GFP marker, and this is Fas3, which labels the very nicely labels the membranes of the epidermal cells. Okay, so it's labeling the epidermis. So everything seems to fit with the idea that dilp is very transiently expressed in the epidermis at the white pre-pupa stage, and that it's needed at this stage for the correction of FA. So is it needed in the epidermis for the correction of FA? Yes, it is. This is shown here. So basically, if we remove specifically with two specific drivers for the epidermis and two drivers for and two RNAis for dilpate, if we remove dilpate from the epidermis, we see that this is sufficient to induce fluctuating asymmetry. And in fact, this is correlated with a reduction of expression of dilpate. So basically, if we reduce dilpate, total dilpate mRNA. So if we remove dilpate with the RNAi using epidermal driver, these two epidermal drivers, this is really knocking down dilpate expression at the white prepupa stage. So dilpate in the epidermis at the white prepupa controls FA. Okay. What is controlling this peak, this very transient peak of dilpate expression in the epidermis? Well, it looks like white pupa is a stage where what we know is that there is a very strong induction of ecdysone production. So of course we were tempted to look for ecdysone receptor signaling driving dilpate expression at that stage, and it looks like indeed ECR, so the receptor for ecdysone, is playing a very important role in driving this peak of dilpate expression. And we have both QRT-PCR experiment here showing that if we knock down ECR using these two epidermal drivers here, this is knocking down dilpate expression. And this is shown here using this dilpate GFP marker again, and a clonal knockdown of ECR signaling using an ECR dominant negative expression here in clones of epidermal cells. What we see here is that if we remove ECR function in these red labeled cells here, we also remove dilpate accumulation using shown using this dilpate GFP mimic. What happens if we remove ECR signaling in the epidermis? We get FA. So indeed, ECR seems to be needed in the epidermis to receive the ecdysone signal, leading to dilpate expression and leading to the control of FA at this transition. Okay, last bit of data. What happens downstream? So what we knew from our previous experiment is that dilpate is acting on the production of ecdysone. We also knew that it seems to be relayed for that through the function of LGR3 neurons in the brain. So now what we're showing him here is that if we measure the circulating levels of ecdysteroids in response to the knockout of dilpate in animals which have no dilpate, we clearly see an upregulation of the circulating levels. This is true at the very late L3, when it's not significant actually at this stage, which is good because this is a time when dilpate is not expressed, but it's significant here in 0. So this is typically a white prepupa, and 2 hours, 4 hours, 6, 8, 12 hours after white prepupa, we see. So we see first an upregulation of circulating ecdysone, and then we see a faster reduction reduction of the peak of accumulation of ecdysone, right? So ecdysone goes up higher, but it's also going down more rapidly, which is probably due to this negative feedback of ecdysone on its own production past the peak. What happens? So this is circulating levels. What happens in terms of targets of ECR, so targets of ecdysone receptor signaling in the discs? So these discs are dissected from either wild type or dilpate KO. And what we see here is that 8 among the 9 targets that we've looked for ECR signaling are actually upregulated. So it means that indeed dilpate is adjusting the level of circulating ecdysone and the level of ECR signaling in target tissues like the developing discs during this period. And if we remove dilpate, we have that effect. So this is done at the white prepupa stage. Okay, so just to conclude rapidly, this is the schematic we have for dilpate functioning and controlling the variability, the developmental variability. Everything seems to take place after the larva to pupa, at the larva to pupa transition, where dilpate is very strongly induced through the action of ecdysone, which is also raising at that time. And the net effect of dilp upregulation is to control, dampen the production of ecdysone, and therefore the level of ecdysone receptor signaling in the developing tissues like the imaginal discs. And this is key to control developmental variability at that stage. Okay, I will finish with something that we have developed in the lab through the work of a very talented postdoc, Roshan Vijendravarma, which is more evolutionary oriented. So the question is, how are these mechanisms for the control of FA? How are they selected during evolution? What are the cues? What are the mechanisms through which these buffering or canalization mechanisms have been evolving? So two ways, of course, according to Charles Darwin. One is the natural selection, and the other one is the sexual selection. And this is very nicely said by Charles Darwin in his second book on the descent of man and selection in relation to sex, which was published in 1871. Sexual selection arises from the reproductive advantages that certain individuals have over others of the same sex and species due to mate choice or competition among individuals of the chosen sex. So in other words, the question that we were asking is, do individuals use fluctuating asymmetry or genetic instability, or developmental instability, sorry, as a cue for the genetic quality of mates? So Roshan very inventively developed a way of generating populations of animals with high levels of fluctuating asymmetry or developmental instability without using genetics, but using microgravity. And the rationale for using microgravity was that in response to many environmental variability, like food, temperature, density, all these things, individuals have developed buffering mechanisms that allow canalizing, that is the right term, the developmental processes. So in order to create, through environmental parameters, to create FA, to create developmental variability, one would need a parameter that normally does not vary, and gravity is one such parameter. So what Roshan did was to use two different devices to modulate gravity, or the sense of gravity during the development of the flies. So this is actually while the animals are developing as embryos and larvae and pupae. So he could rotate them vertically. So of course, then the gravity is actually varying during the rotation as compared to the horizontal rotation, which serves as control. And he also used these random positioning machines, RPMs, which were lent to us by the European Space Agency, which allow completely random positioning of the samples. And therefore, the animals have a completely perturbed sense of the gravity, because they cannot expect in which direction they will be moving, and therefore they cannot really habituate to some kind of pattern of microgravity variation. And so in these two cases, so vertical rotation and RPM, what he observed after developing animals in these conditions is a very high increase in FA index, and the horizontal here serves as a control as well as the still condition. So that was very surprising, and we're still working to understand why we get these effects, and Roshan has been doing a lot of experiments to try to understand what are the consequences of microgravity or perturbed gravity sensing on developmental parameter and developmental stability. And it's an axis of research per se. But he could use these populations to ask the question: how are these individuals with high noise? How are they behaving in mating choice assay? So this is the assay that Roshan developed. It's a very elegant assay where he's picking randomly picking two males which have been undergoing microgravity effect. So these males have, in the population of these males has accumulated high level of developmental variability. But we don't know anything about these two males in particular. What we know is that they might have different levels of variability, right? But we have no idea. They've been picked completely randomly. And they are actually mated with one, I mean, they've put for mating assay with one single female, which is a still female. And so what happens after a while is that after courtship takes place, is that one male succeeds in mating while the other does not. And so we have a winner and a loser, a winner for mating and a loser for mating. And now if after Roshan made quite a number of individual crosses, more than 29 vials of individual crosses, and picking every time, the loser and the winner male, he could reconstitute a loser population and a winner population. And then he could examine the FA in the loser population and the FA in the loser population. And very dramatically, what he sees is that there is a very strong increase in the variability in the loser population, so the males that couldn't make it mating with the female, whereas the winner males have absolutely no variability. So in other words, this mating assay is able to select for males which have, the mating males which have very low developmental instability. So that was a sign that indeed, through mating choice, through sexual selection, the males with high FA are counter-selected. Okay. Since Roshan has done a lot of experiments, and we were preparing a report on that, but I don't have the time to go through for all the experiments he's done. But this is one of the interesting experiments. Is the woman choosing the female choosing? I mean, we know that the woman is choosing, in fact. But what about the female? Is this selection going through the female, or is it going through a competition between the male? So this is one. I mean, the two possibilities are actually equal. What he found doing this experiment here is that it's probably going through female selection. And what he did here was to use selected populations of females for either monogamy or for polygamy. So and forced monogamy females have been crossed individually with one single male for a high number of generations. So they are not used to polygamy, right? And on the contrary, the polygamy is the control here. And if we do the same mate choice assay using these two different selected female populations, what we see is that this is the control. So this is equivalent to what I just showed to you. With polygamy females, polygamy behaving females, the females are able to choose the males with low FA. So the difference between male and female is low FA and high FA in winner and loser is very clear. Whereas in monogamous females, if we put these monogamous females with the two males here, they don't actually exert their capacity to choose between the two males. And this attenuates the difference between the loser and the winner FA. Okay, so we think that one component in this selection here is actually coming from female choice, but probably not the entirety of it. What Roshan did was to use another way of creating FA, which is this genetic way I just mentioned to you. So he used dilp KO animals. We know that in a population of dilp KO individuals, there are animals with high FA, animals with high variability, animals with low variability between the two wings, but not only the two wings, right? It also concerns all sorts of different traits. And so if we take two males randomly picked in this population of dilp KO over KO, and we do the same mate selection assay, we get exactly the same result. So basically, the winners have much lower FA than the losers. So again, this is the same data, but with a very different way of generating FA. So either the genetic way, like this, or the environmental way. In both conditions, what we show is that the sexual selection is very efficient at in fact selecting for low FA individuals. Okay, so I don't have the time, I guess, to go much further. I'm probably late, yes, I am. And I would like to finish by thanking people who have done the work: Laura Boulan, Roshan, Dalmiro, Obregon, Sunita, Kaula, and Fabienne. And these are the two people who initiated the work, Julien and Dieter, our collaborators, and the funding bodies. And thank you very much for your help, and all my apologies for being too long. Thank you.
Thank you so much, Pierre. No problem. We are still okay with the time. Let me see if I can put this both.
Can I stop the sharing? Maybe
Yeah, I think
sharing?
it's fine. Yeah, I think it's fine.
Okay.
Okay, so we have some questions. I cannot put this both. Let me put you on screen. So we have a few questions from the audience, and I will read it to you. Okay.
Sure.
Bassem Hassan asks, "Hi Pierre, two questions. One,
Hi,
is there
Are you
a
in Portugal now?
hierarchy of privileged discs, or are they all equally able to delay population if injured?" And the second question is, you show that DILP 8 is not necessary but sufficient for the timing under normal conditions, but is it required upon injury or regeneration?
Right. So other discs, yeah, this is a question that we have not addressed. I mean, we should really address that because this question is actually coming back very often. No, we haven't looked at the effect of altering growth in other tissues, whether it's equivalent for. Right. For me, I mean, we see induction. We've seen induction. It's not completely true, actually. We see DILP 8 induction upon pinching larvae. So it's sufficient to actually to pinch the larva at a given, in one specific region to see a strong induction of DILP 8. And so if we pinch other discs, I mean, we see induction in the other discs as well. So we haven't done it genetically, but I'm convinced that it should work with other discs as well. I mean, the pinching, the physical alteration, the physical stress is sufficient to induce DILP 8, which is one difficulty of working with these animals. I mean, we want to be very gentle. If we pinch them too hard, there is a burst of DILP 8 expression. So, yeah. So I would say yes. I mean, we don't have the genetic data, but I would say yeah, it's probably true for other tissues, right?
Very interesting.
Second aspect was whether so it's not needed for normal development if everything goes okay, but if we alter growth as soon as we alter growth, in some ways in developing tissues we have a burst of DILP 8 upregulation, and this is actually what causes a delay. Yes.
Okay. So we have another question from Savraj Griwal. I hope I'm saying it correctly.
Well, I didn't realize it was an international meeting. Yeah, this is amazing. Yeah, this is great. Yeah.
Is the LGR3 receptor? Oops. Where is it? Okay. Is the LGR3 receptor expressed only on the neurons connecting to the PTTH neurons? Are the DILP 8 LGR effects mediated exclusively through ecdyson, or could DILP 8 also act directly on peripheral tissues?
Yes. So, well, this is a very vast question. I'm showing you a subset of LGR3 positive neurons. As I mentioned, there are actually plenty of them. And so far, well, there is this very nice preprint from the Gontijo team, which shows that another subpopulation of neurons are actually involved, and people have probably heard Alison talking about that before, which are involved in a very specific contraction system that takes place during the larva to pupal transition, which is really necessary to prepare the pupal case. So, and this goes through a second population of neurons, which are LGR3 positive. So we think that DILP 8, again, in this configuration, I mean, Alison thinks that DILP 8 goes through the function of LGR3, but in a subpopulation of neurons. And we could probably think that there are many other subpopulations of neurons which use DILP 8 as a trigger to do other things, right? So we don't have a very clear picture of that for now. So it's quite possible that DILP8 acts on LGR3 through LGR3 in other neurons to conduct other functions, either during this stage of development, later during development, at the end of pupal development during adult. There's a very nice paper showing that that LGR3 is needed in the ovary to mediate. LGR3 is needed immediately. Is expressed in ovarian cells to mediate DILP8 expression, DILP8 effects on fertility. So, but again, it's going through LGR3. So my feeling is that in many cases where we see DILP8, it's actually linked to LGR3 for functioning. It does not. We cannot really exclude, and Alison showed that some time ago in a paper that it could work through the insulin receptor itself, and I don't think we can exclude completely that. We don't have we don't have clear evidence for that, right? So it's a DILP. Whether well, I mean, I don't really think it's a DILP. In fact, I rather think it's a relaxin, and this is a discussion. It's like some kind of a semantic discussion. I don't think it's very important, but in fact, yes. I mean, I think we all agree, people working on the DILP8 field, that it acts more like a relaxin than like an insulin peptide, like peptide. But probably it needs LGR3 for at least for all the functions that have been described so far. Okay. And in terms of neurons, yes, I mean, lots of black boxes. There are plenty of LGR3 positive neurons. They might not all need DILP8 to function, to exert their function. So that's another possibility.
Okay.
They could be promiscuous and work with other ligands. Yeah.
Okay, Pierre. Thank you so much. So, so we have a lot of other questions. I'm going through the questions as they are voted. Okay, so the first now, the top question is from Carlos Ribeiro. Very cool data on the impact of FA on mating competition. What is the sense used by the female to choose? Is it vision or is it hearing? Asymmetry might lead to problems in singing as well.
Exactly. So, yeah, this is a vast program, Carlos, and we're just starting exploring this, but we do it, and Roshan does it with a lot of energy. So basically, we've done experiments in the dark. We've done experiments with white mutants. So if I don't have the time to show all this, but it looks like the visual cue is important. So basically, in white mutant animals, we lose quite a big part of the selection, so of the difference in FA between the loser and the winner. So meaning, but again, I mean, this is very difficult to draw a conclusion, because the white mutation is, of course, changing the pigment and the vision, but it might be doing something else, and this is the difficulty we have with working with mutants for this kind of assays. But if we put animals in red light, we also reduce the difference in FA between the loser and the winner. This all suggests that the visual cue is participating in that. Roshan has cut the antennas, to see whether the smell could be important, and we are doing all sorts of measurements on rotated males to see what kind of hydrocarbon profile they have, whether this hydrocarbon profiling is changing, and whether we can do a correlation between loser profile and winner profile. So this is ongoing. The sound, of course, if the wings are not perfectly symmetrical, you could imagine that this would have an effect on the sound. So Roshan is participating in a collaboration with people who know how to record, the sound in stereo, so they can actually record the left wing sound and distinguish it from the right wing sound. So this is what we're doing now, and again, I mean, comparing the left and the right songs, basically, because they alternate during the courtship. We probably can do some correlation between the loser behavior and, the loser trait and this kind of behavior. So all these things are undergoing. What else is Roshan doing? Yes, so Well, Roshan, of course, is very interested in trying to understand whether this can be transmitted to the next generation. So he has experiments ongoing on that aspect as well. And this is so, yeah, right. We're right in the middle of that. Thanks, Carlos. This is
fantastic. Okay, so now we have a question from Alison Gontijo. Hi, great talk, Pierre. You show a high peak of 20 HE in DILP8 mutants than wild-type mutants at T0. However,
a peak of
ecdysone of 20 HE.
Okay.
In DILP8 mutants than wild-type mutants at T0. However, you and us have shown that DILP8 and LGR3 mutants pupariate around 3 to 4 hours before wild-type animals. I think work that the ecdysone peak occurs at 4 hours before T0 in wild-type animals. Does this mean that DILP8 LGR3 mutants are pupariating at the time of highest ecdysone titers instead of doing it around 3 to 4 hours after, when these levels have reduced a bit? How would this affect your model?
Well, so I guess the question from Alison, I guess. Yeah, because it's a very specialist question, right? So, yes. And I would need to reread it to make sure I understand it fully, because it looks complex. What I showed you is that animals, actually the curve I'm showing you concerns animals which are synchronized at the wide prepupa, right? So whether they have 2 hours, this time is occurring 2 hours early or not, should not make a difference in the analysis that we are doing. So basically what we see is that in a DILP8 mutant at wide prepupa, the level of ecdysone is slightly higher, as we would expect from the role of DILP8. But indeed, and this is all I can say, I would really need to read through the question again to be more precise, but indeed, we cannot say whether this wide prepupa is taking place slightly before or slightly after in these different categories of individuals, right? For us, yeah, I mean, this advanced preparation, this is something that we've been banging our head against. In some cases, we see this slight advance. In some cases, we don't. And so I'm not completely sure at the end what is the reality. I mean, I would have to discuss with people in the lab who have done the work. But here, again, I mean, we select animals at wide prepupa.
Okay, so I think I'm gonna just read you one last. We are a bit over time now. So a question from Clotilde Quimon. Some species of Drosophila relies more on olfaction than sight for mating. Is there any natural variation of asymmetry between species or even across melanogaster strains?
Well, that's also a very interesting question. So basically, we would like to, since we're using non-genetic ways of creating a FA, we can use a variety of different species for that, and this is what of course Roshan is an evolutionist, and she's very interested in comparing from species to species and to see whether this sexual selection that we see against variability is something that also operates in other species, and in particular would be very, very interesting to see what happens in species which have different senses of which use different senses for mating, for instance. Yeah, typically. Yeah, that would be really. I mean, that would be absolutely great. Yeah, to do that. Yeah, it's a great question.
Okay. Yeah. I'm so sorry, but I don't think we will have time for reading any further questions. Thank you so much, Pierre, for joining us today. And thank you everyone that joined us. I hope to see you all in a couple of weeks in our next seminar. And Pierre, I will see you in maybe a few minutes in the Zoom room. Okay?
Sure. Okay.
Thank you so much.
Thank you, everyone. Ciao. Thank you for your great questions. Bye, everyone.